I open this academic session in which Mr. Francis Morgan will defend his academic thesis, Dynamic Bond, Rational Design of Dynamic Covalent Soft Matter and Application Tissue Engineering. I welcome you and wish you lots of success together with your paronyms, and I also welcome your supervisors, Professor Moroni, Dr. Baker and Dr. Bauer, of course the members of the opposition, online and on site, whom you will meet later, and of course your colleagues and friends and family on site and online. But before we start the opposition, I would invite you to present a presentation about your work which has been done for the PhD.
Thank you, Pro-Rector. Thank you, Corona, for being here today, and everyone else for coming. So I would like you first to consider the human body and the types of materials specifically that we use when we are trying to repair it. If anybody knows somebody who's been through a hip replacement, for example, you might know that those are generally made out of titanium with a plastic cap. Funnily enough, plastics and metals do not behave very similarly to the way that the human body does. They do not bend and flex and stretch or repair themselves the way that our body does. So in the last decade or two, we have seen quite a lot of development of what are called dynamic hydrogels, which I'll come back to in a second, which are materials that are much more reminiscent of the human body. They are primarily made up of water, they can be a lot softer, we can control a lot more about their stiffness, and generally they are leading the way forward for developing materials that are much more adapted to repairing the human body. So the goal of this thesis, in large part, was to push this development forward and create new tools and materials that we can use to replace different elements or aspects of the human body for medical and therapeutic uses. So for the non-scientific audience out there, I'd like to cover a little bit about what exactly I mean by dynamic covalent hydrogels. So a hydrogel, simply speaking, is a network of polymers and a lot of water. Normally more than 90% water. And a polymer, in this case, is connected with what we call a crosslink, which is a chemical bridge that joins these together. So to build on that a little bit, a polymer is simply a long chain of repeating units. So these chains can be longer, they can be shorter, we can have different types of units, we can have very similar units, and these units can have specific functions associated with them. So specifically dynamic hydrogels, what I mean when I say dynamic is that these chemical bridges or these crosslinks that we are making are reversible. So they can be in a state that you may think of as on or bound, or a state that is off or unbound. And it is this reversibility where these crosslinks can go from being in one state to the other and back again, which is very interesting and what we are trying to leverage throughout this work. So how is this useful and what do we leverage? Well, we can define the way that this reversibility behaves, the way that it goes on and the way that it goes off. We can define how fast this happens, we can define how fast it unbinds, and the important parameter that I'd like you to retain for the next 10 minutes is this equilibrium constant, which is the ratio between how fast it binds and unbinds. And so, if we consider this in the context of a network, then this equilibrium constant is going to tell us exactly how many out of the entire population of these crosslinks are bound based on how fast they bind and unbind. And so I've got an example here that we've prepared where you can see a hydrogel that is made with a low KEQ crosslink and a high KEQ crosslink. So the higher the KEQ, the faster the relative binding or on rate is, the more crosslinks there are and the more rigid or mechanically robust, as you can see in this photo, the network is. So the reason that this is really powerful is that it creates a link, a direct link between this chemical or molecular property that we can quantify and this macroscopic behavior of these hydrogels when we have them in our hands. So one of the first things we set out to do in this thesis in chapter 2 is build upon this knowledge. So since this is a relatively recent field, most examples use this relationship in a qualitative manner, and there are a limited selection of available crosslinkers that have known and defined binding and unbinding rates and equilibrium constants. And so in chapter 2 what we did was we took a selection of common and less commonly used polymer motifs as well as cross-linking functions, and we quantified them. So this graph here, or this plot, shows an example of how when we start reacting these polymers and cross-linkers together, they can exhibit different rates. So in this case, a steep slope is a fast occurring reaction, whereas a less steep slope occurs more slowly. And similarly, at the end of this graph, you can see that the different positions are going to correlate to different equilibrium constants as they balance out with one another. So quantitatively, that leaves us with a large number of numbers which we can use. But what we want to do with those is take them a little bit further. We create this small library of reactivities. We've quantified them. What can we do with them? So the example for this that I'd like to give you today is we wanted to consider what happens if we start mixing them together, if we add a little bit more complexity to our system. And so what happens if we take a high KEQ system and we add some of the lower KEQ crosslinker to it to get a mixed system? And so here we developed some mathematical models, and we did some calculations, and we get a scary looking graph like this. But the important piece of information that you need to take away is that this black region is telling us that it is predicting a softening of this hydrogel when we create this mixed system. Now when we validate that experimentally, we can see that in fact that holds true. And that our model in this particular case has some predictive value. So that's particularly useful because it demonstrates that by going to this extra effort to quantify them, to obtain this precise knowledge of rate and equilibrium constants, we can not only identify trends in the reactivity of these different systems, we can provide more systems for people to use, we can translate the model data into practical systems and also identify novel and perhaps unexpected ways that these systems will behave together. Now in chapter 3, we wanted to take that a little bit further and not look just at how these motifs react together, but what if we take this chemical bridge, this crosslinker, and we also change its structure and composition a little bit? What if it's really long, or if it's really short, or if it has multiple arms? Now, we got some quite interesting results out of that, but one of the main hypotheses that we came away from this work with is that we think that there is quite a large difference in the relative degree of intramolecular cross-linking. Now, what I mean by that is that these bridges, instead of forming between two separate polymer chains, are forming between the same polymer chain, creating a loop within itself. And that has a dramatic impact on the resulting mechanical properties that we obtain from hydrogels. So with a lot of fundamental work under our belt, let's take a look at a little bit more of a practical application of this. So in chapter 4, we set out to make a dynamic bioink. In this context, a bioink is a hydrogel that is loaded with cells that we can also put in a 3D printer, for example, in order to have a spatial arrangement as well as a composition of this material. And so if you look at the same example again of the softer hydrogel, this was made with what are called hydrazone crosslinks. And if we look at the stiffer example, which was made with what we call oxime crosslinks, we can also mix them together in much more varied compositions at the very beginning in order to achieve intermediate behaviors. Now, that is particularly useful because we cannot necessarily control the equilibrium constants. We simply measure them and find out what they are, and this allows us to access stiffness or mechanical regimes that are intermediate between them. This is particularly useful because in this example here, if we look at some of the mechanical properties of a human body, which themselves are quite diverse, we can see that our hydrogel system here, as we varied the composition between the two crosslinkers, was able to capture some of that difference in behavior, which is encouraging because it means that we have the possibility to target some of the mechanical properties of specific human tissues in order to help drive the growth and eventual regeneration, hopefully, of these systems with these materials. Now, of course, this is the mechanical aspect. The cell aspect is also important, and I'm not going to go into too much detail, but comparing the two extremes of these, we can see that in the softer system here, we can drive a rounded cell morphology, whereas where we use the stiffer system, we can drive a more spread morphology on these cells, indicating that we can use this difference in chemistry alone to achieve significant impact on the way that cells respond and develop in these systems. So as we move on to chapter 5, we wanted to ask a slightly more different question. We know that we can control the mechanical properties of our hydrogels. We know that we can use them to direct cellular development. But we wanted to understand a little bit more how that cellular development is directed. So the question we wanted to ask here is, well, do cells see the same mechanical properties that we measure with our machines in the lab? So the important piece of contextual information here is that cells interact with their surroundings by using what are called integrins. And one of these integrins is called RGD. You can imagine them as small hooks or feelers that go out from the cell and are able to tug and pull on the environment that they're in order to get an idea of how or what its mechanical properties are. And so here we set up a hydrogel network, slightly different compositions, but the key feature of this is that we could introduce different types of RGD, or RGDs that were connected to the network with a slightly different chemical motif, one being dynamic and reversible, the other being irreversible or static. And we wanted to see if the cells saw a difference, because when we add these motifs to our system, they do not impact the mechanics at all. We as scientists measure a single set of mechanical properties. Do cells see a single set, or do they respond differently? And as it turns out, they respond quite differently. So in blue we have the dynamic RGD, in orange we have the static RGD. When there's no RGD, they do not adhere, and we can see that in this again, the cellular morphology changes quite dramatically, but we do not measure any difference in the mechanics. And so this allows us to identify the need for different tools or different approaches to studying these sort of materials in order to really understand what mechanical property or what scale of mechanical property the cells that we want to put inside of these materials are going to respond to. We did go a little bit further in this study in chapter 5 to quantify this at a genetic and microRNA level to further support our findings. Now finally, in the final chapter, chapter 6, we wanted to make a new type of material, a synthetic alternative. All of the polymers or the hydrogels we've made so far have been made with alginate. Alginate that has been isolated from seaweed, a naturally derived biopolymer. The naturally derived polymers can be great, but they do have one major drawback. They are generally very heterogeneous. That means that these polymer chains that they are composed of tend to vary somewhat in length. They can vary slightly in terms of the arrangement and composition of their units. And while they can be very functional, that does preclude us from having a very highly defined chemical system which can be attractive for certain applications. So in chapter 6, we set out to design a well-defined synthetic copolymer using multiple different subunits so that we can mix and match different functionalities, that we can retain solubility while also allowing us to perform dynamic covalent cross-linking to form the hydrogels with them. And so we achieved that. To give you an idea, these are some of the chemical structures of the two subunits that we used, and we were able to form these copolymers with a variety of different compositions and study and show how this different composition allowed us to access different behaviors in terms of how fast they gel, what type of mechanical properties they have. So today I hope that I've been able to show you that through the quantification of these molecular rate and equilibrium constants, that we are able to model and predict some of the resulting material properties, that the mixing of these dynamic cross-linkers to create a little bit of complexity to the system was a very viable and useful tool in order to access a wider range of mechanical properties, that we've also uncovered some new questions about how we measure and study these sorts of systems, the mechanics of these systems, how they interact with cells. That will be a very solid starting point for future work and studies as the field moves forward. That we created a synthetic alternative to facilitate the well-defined chemistry so that we can perform certain types of studies in the future, and that overall, taken together, these and the work within this thesis has provided new tools and insights and a strong foundation where we can develop advanced soft dynamic biomaterial systems for the future. So thank you very much for listening, and I pass the word back to you, Pro-Rector.
Thank you, Mr. Candidate, for an interesting lecture. We start now with the opposition, and the opposition is opened by the chair of the assessment committee, that's Professor Oru, and he's professor in bio-based organic chemistry in the University of Maastricht. Professor Oru.
Thank you, Mr. Rector, for giving me the opportunity to discuss with you some aspect of the thesis that you have produced, which is, by the way, impressive and elaborate. It was a pleasure to get into the topic and read the whole thesis. So my question actually is related to chapter 2, at least the first questions. And that chapter is, well, starts off with explaining that your aim is to build a structure-activity relationship that amines display with amines and aldehydes, between amines and aldehydes. So if you then look at the choices you make for performing such a study, especially first perhaps looking at the amine nucleophiles that you have chosen, and then you have in table or in figure 2 listed the ones that you have picked. And especially on the amines, then you show that you, let's say, gradually change functional groups, for instance, a carbon into an oxygen, etc., which have impact on the chemical properties, of course, of the amine that should form the imine. And you also list there the pKa values of these different molecules. So if I now look back at your original aim, this really focused more on the influence of the hetero or the atom next to the nitrogen atom, influencing the pKa value and therefore also influencing the equilibrium of the imine formation. For a, let's say, more comprehensive or more maybe a study of a real structure-activity relationship, maybe I put it a bit more provocative, you of course need to also look at a more subtle changes. So, and the question from me is now, why did you pick primary amines, for instance? The first amine that you have in the table is a linear primary amine. Why not a secondary amine? Why not a cyclic amine or a conjugated amine, which would have also offered quite some information on this equilibrium next to the ones that you have picked already?
Highly esteemed opponent, thank you for your comments and your question. The rationale behind the selection there was, well, the primary amine was intended as a control, a starting point. It's a very known quantity that a lot of people can use as a starting point to then understand the rest of the changes that we're making. Beyond that, we wanted to make sure that these were functions that ultimately could be used as crosslinkers, so attached to the end of, for example, a peg chain to then form a hydrogel network with, which can be a little bit more challenging for as we added too much complexity to these functions. And the most commonly used crosslinkers in the field are based upon all of the previous work in bioconjugation are the hydrozones and the oximes. And so we also chose the series such that we could go through the hydrozone and the oxime to make sure that we included both the common and well-known crosslinkers, as well as subtle varieties around them that we explored to highlight not only, hey, there's some actual other options that could be potentially interesting in these types of projects as crosslinkers, and also underscore how even changing a single atom in one of these structures, which, yes, are primarily the heteroatoms, alpha or beta from that terminal amino group, can have a significant impact on the reactivity, this molecular property, and one way of looking at that is, of course, the pKa, and then ultimately how that impacts the resulting mechanics that we get, which is, again, 1 degree further removed from that chemical design.
Mm-hmm. Okay, yeah, I understand, and it's also, let's say, understandable that you have to, let's say, you cannot test everything, so otherwise the whole library would become perhaps too big. But in relation to it, I also was triggered about a remark that you made on page 39, which states, after testing several aliphatic aldehydes, So now we are talking about the aldehyde, let's say, component. Only isobutylaldehyde and cyclohexane carboxaldehyde prove stable under the aqueous conditions without complex secondary structural forms. So then, in combination with the question that I asked just now, you choose, let's say, this branched aldehyde as the small one as a starting point, and then a very big alginate-based aldehyde and this synthetic polymer. But these are all aldehydes, and as you know, probably, the alpha side of aldehyde, also by the way of ketones, that contain an acidic, a relatively acidic proton, which causes perhaps these, as you call them, complex secondary structural forms. These are probably aldol type reactions. So is that true, and did you observe these aldolization? And depending on the amine that you then choose, you might be able to, let's say, direct this aldolization or Manech type chemistry.
Short answer, please.
So the NMRs for those are in the supplementary information. We made sure to verify that we only had a single aldehyde population for the synthetic copolymer and the small molecule, because otherwise the analysis, particularly of the kinetics, becomes extremely complex when we're dealing with the isoform conversion between aldols and aldehydes. The alginate we chose because it is extremely commonly used, but strictly speaking, there are no free aldehydes, also shown by the NMR, because that exists primarily in the stabilized hemiacetal form for the neighboring alcohols on the adjacent uronic acid units.
Okay. Well, thank you.
Thank you very much, Professor Ori. Now we come to the following opponent, who's also a member of the assessment committee. That's Professor Hakeng. He is professor in biochemistry in the University of Maastricht, and today he has also accepted the role of secretary, for which I'm very grateful. Professor Hakeng.
Well, thank you, Mr. Prorector. Dear candidate, I would like to first compliment you on this chemical masterpiece. It's really a very nice chemical biological piece of work, and it does not only contain experiments, it's basically work of concepts, and I congratulate you on that. It has led to many outstanding papers with a stunning amount of citations already, and of course these compliments also apply to your promotion team, Dr. Baker, Professor Mahoney, and Dr. Bauer. Dynamic is the focus of your work, in which the versatile imine bond is at the center of the stage. And first, I would like to ask you a little bit of a philosophical question. To which part of your work is the word dynamic the most appropriate? Is it the many chemical functionalities and experimental conditions leading to combinations of rate and equilibrium constant with a dynamic range of over 3 orders of magnitude that allow you to dynamically design hydrogels with various properties? Or is it the intrinsic dynamic nature of the imine bonds that allow you to make, break, exchange the imine bonds at will in already fabricated hydrogels? And you can't say both.
Highly esteemed opponent, thank you for your comments and your question. That's quite an interesting choice that you have put in front of me today. I think at this stage, for the development of the field, it truly is the dynamic toolbox available to us that is the most important right now. However, I believe that the intrinsic nature of it is what's going to become a lot more important once these tools are in place in order to achieve highly specific goals.
You speak in wisdom beyond your years. I tend to agree. I think in this aspect you're much wiser than I am, but I think that indeed, it's how you say it, the future will make the second proposition more valued for dynamics than the first. So talking about that, you basically challenged us during your presentation to consider the human body. I would like to ask you to do the same. So how would you engage, and there are many possibilities, how would you engage in the clinical applications of your versatile toolbox of making hydrogels and thinking about preceding of autologous gels, seeking out cells in the body? How would you do that? talking about arresting cells with integrin ligands, or maybe seeking out gradients for chemotaxis. What would you think would be the optimal strategy? And also maybe, you probably have the rest of the time for this with my interruptions in there, how would it be favorable also to release structure, hydrogel structure by competing ketones in the body to let it fall apart if there's enough cell ingrowth and stuff like that? What's your opinion about that?
That's a fascinating question. Again, it being a tool, the applications ultimately are all of the applications that we can imagine, which is quite a lot. In the first case, as with any clinical translation, I believe the first approach needs to be the most pragmatic. What is going to be easy to develop, safe to develop, easy to get a regulatory approval for, and easy to use for the clinicians. So in that case, we can imagine scenarios where we have simply an injectable material that they can insert with minimal trauma to the candle or to the patient that can have some positive therapeutic effect that is currently unavailable. So one prominent example of that is injectable hydrogels for spinal cord injury to promote the regeneration of severed nerves there. You can engineer triggered release, dynamic release, either through a self-added competitor, a degradation component within the gel that will also lead to a slow release over time, or the in vivo competition using biological components secreted by cells.
And this would not involve cells? Seeding of cells in the hydrogel?
Not necessarily, no. And initially not so much, mainly because I would argue the regulatory hurdle to not have cellular material in it is much lower, which enables us to get something to the clinic much faster. And once something becomes established clinically, it's a lot easier to build upon that and add more complexity later on. Now, in terms of 15, 20, 30 years down the line when we have these systems and how best we can leverage them, I think one interesting question to try and answer is how we can regulate the development of neo-tissue following injuries, or how can we use that to supplement the regenerative process that exists within the human body already. How we can use those dynamics to respond to changes that the body creates. Now, whether that's chemical, biochemical, mechanical, there are lots of triggers that can be built into these. The other very nice advantage of these systems is that they can be designed in such a way as that to be orthogonal to a lot of the other responsive strategies that we see within hydrogel systems. And I think that one of as the healing process occurs, naturally there are many changes in the mechanics of the matrices surrounding that. And I think that's going to be a really interesting area down the line where these types of materials can help guide, regulate, and respond to those changes.
And in the end, would you like to get rid of the hydrogels in the body, or would they stay?
Ideally, they shall degrade and be eliminated, or dissociate and be eliminated. The advantage of dynamic systems is that we can undo all of these crosslinks. We show in Chapter 6 that we can make a hydrogel and then completely remove said hydrogel by adding a competitor. And the same process could be engineered to happen within the body such that they can then be eliminated and leave behind a hopefully pristine native tissue.
Okay. I have a very short question. Mr. Prorector? Yeah. So have you considered the influence of endogenous ketones in our body that influence your hydrogel stability and structure, such as, for example, typical male molecules, acetaldehyde and testosterone?
Yeah. We have considered them in our discussions. They have not been considered explicitly within this thesis yet. That is very much a question to build upon what we have currently. One example I can give you, for example, is even when we do cell culture media is able to occupy a certain fraction of aldehydes if they are left in a stock solution, for example, for an extended period of time. And so that already impacts the relative or the apparent reactivity within these systems. And so the behavior that we observe and that we can demonstrate here ex vivo and even in cell culture conditions will never quite be identical to what we observe within the human body. So those will need to be taken into account as we develop therapeutic treatments. But they can also be beneficial once we have a better understanding of exactly how they behave.
Exactly. Well, I'm very happy with your answer, and actually I'm a little bit impressed. I give the word back to the prorector.
Thank you, Professor Keng, for your position. The following opponent is also a member of the assessment committee. That's Dr. Diaz, and he's online, and his expertise is material biology interaction at DSM Firmenich in Geleen in the Netherlands. Dr. Diaz. You are muted.
Thank you, Mr. Prorector, and good afternoon, Mr. Candidate. So it was a pleasure to read your thesis, very much thought-provoking. And my questions are really devoted towards applying these systems. How can we translate these to the clinic? And I'd like to begin with the first one, which is the overall research thrust. When you look at dynamic interactions, they've been the benzene tricarboxamides, UPs, uradipyrimidones, cucurbitryls, and a number of other interactions. When you're looking at translating these systems, it is important you consider that they work each time, all the time, every time. That's the reliability that's demanded for going into such medical applications. So what's influenced your choice in terms of controllability? And more importantly, have you considered the biological fate after they've been used?
Highly esteemed opponent, thank you for your comments and your question. Oh, there we go. Regarding the consistency of all of the results and the behavior of these systems, that in part was one of the elements that we address in Chapter 6 by providing a synthetic alternative where its composition and therefore behavior can be very well defined compared to perhaps batch-to-batch differences, sourcing differences that you see in native materials. In terms of the nature of the function itself, dynamic covalent chemistry, while all dynamic chemistries are sensitive, the dynamic covalent chemistry is potentially less sensitive to the physiochemical properties compared to, say, the UPs or cucurbitryls, for example. They will each have their own pros and cons, but in the context of the human body, considering the sensitivity of each of those dynamic functions to pH, temperature, salinity, and other physiologically relevant conditions and how they vary, I think the dynamic, or I believe the dynamic covalent chemistry is one of the more robust among them. And, sorry, the second part of your question was regarding the biological function, if I'm reading your lips correctly. Sorry, you were muted.
Biological fate.
Fate. Yes. So alginates are already quite well known to have an acceptable biological elimination pathway. That's also one of the common advantages cited for the natural biopolymer systems. For any synthetic alternative, including the one that we prepared, that would of course need to be studied. One of the advantages of the dynamic systems or the dynamic covalent chemistries is they can be reduced or returned to their individual polymers. And that provides access to certain elimination pathways within the body which tend to be favorable compared to a more macroscopic construct that cannot be broken down to its component parts again. It is, of course, possible to build in further responsive units. We could consider having some sort of triggered, whether it's by far infrared or another pH change or added molecule, to break down a polymer into oligomers to further accelerate and facilitate that type of elimination. Overall, these strategies should be applicable in principle to most types of dynamic chemistry if we're talking about a multivalent macromer as opposed to telechelix type structures. I definitely understand the need for for this work and both the composition, reliability, and elimination, but I do not believe either of these are significant obstacles to the implementation of these types of systems in the coming years.
Thank you, Mr. Candidate. That's clear. And it will require further penetration in terms of post-implantation therapy if you're going to manipulate it in that way. So my second question is really devoted to what I regard as the most impactful part of your thesis, which is the density functional theory calculations, because these arguably contribute to the field And as we deal with biological and complex systems, going down the in silico routes is going to be important. Could you share with us what are the limitations, what were the assumptions of these calculations, and how you see them being overcome as you look to using this as a research tool, a thinking tool, a idea generation tool going forward, devoted to complex biological systems?
Yeah, the DFT calculations we performed were very interesting. It was a new experience. It was incidentally based off Mark's work from a paper that he put out on the structure-reactivity functions a few years prior. And the main assumptions that we make there are that all of the changes that we're making, these incremental changes to the terminal amino groups, do not result in any significant change to the mechanism or to the relative difference in energies of the transition states from the products that we are making. In light of the results that we got, I believe our assumptions there are a little bit too generous, given the weak correlations that we did observe between our results. However, the fact that we still managed to get weak correlations despite perhaps having made so many assumptions is itself encouraging that as a tool, if adapted properly for this set of complex systems, can be very powerful. And if we go a little bit further and look at examples of the DFT calculations, again, I can give the example of Mark's paper. Most of these will use a substituent that is being changed, which will then impact how the primary reactive function behaves as a consequence. In our case here, we have made significant changes to the primary reactive function by changing the heteroatom directly, which I believe contributes in a significant part to the deviation or the lack of strong trends that we observe as a part of these results. If you look through the supplementary information, you can even see in some cases the optimized geometry that we have for some of the amine products will switch between cis and trans, which would imply that it is possible that there is a difference in terms of the sort of lowest energy mechanistic pathway in one or two of those examples. So while our results were... Yeah.
That's a very clear answer. I'll just jump to the last question, which is chapter 5, where you modified these double network hydrogels with a cell binding domain and saw the greatest effect.
We are running out of time, so we leave that for later on. And thank you for your opposition. We now go to the following opponent, and that's Dr. Paes, and her expertise is developmental bioengineering in the University of Twente. And thank you for coming all the way from Twente, albeit online. The floor is yours.
Thank you, Mr. Prorector. Dear Mr. Candidate, so congratulations for the great piece of work. I read it with a lot of pleasure, and I find it particularly interesting how you span across many different techniques, and how you put like a lot of effort in connecting molecular reactivity with mechanical properties. So congratulations for that. I'd like to start with chapter 2. And can you, so when I see it, what for me is figure 3 in your chapter 2, can you comment on how K-1 was calculated?
Highly esteemed opponent, thank you for your comments and your question. All of the K1s and K-1s, so the forward and reverse rate constants, were calculated by applying a kinetic model, the solution to the rate equation for a bimolecular equimolar reversible reaction occurring, and that fitting was done using a general fitting program, Origin, and that gave us our K1s and K-1s. As a result of the K1 and K-1, we then calculated the KEQ as a result.
So, but I see in that plot, so you are showing the evolution of the reaction. So that means the consumption of the amine. But where is exactly this the experimental point that I should connect with K-1? Or is the fitting of the equation the most important thing?
So the K-1 is going to retard, to a certain extent, the evolution in the amine that we observe. And that retardation is effectively being quantified by the fitting model that we apply to this data. So yes, I would say that the primary tool that is used is the fitting that we perform. The observation of the trend of the amine over time will tell us qualitatively if it is very fast, if it is very slow. That's very hard to translate visually into the magnitude of the K-1.
Thank you. That's clear. So, and then I'd like to ask you about, so in the conclusion of that chapter, you mentioned that the model that you nicely developed now can be extended and be applied for different situations where we can also have like dynamic interactions. So you also mentioned the case of extending this to non-covalent interactions or to different supramolecular situations. But here I would like to ask you like more generally, because I think the measurements that you have done are related to a kind of dynamic chemistry that's, let's say, comfortable from the timescale point of view for the measurements. But how will you extend this model when now we are addressing very fast dynamic interactions?
So the model we're referring to, or that we use in the end of chapter 2 there, uses the equilibrium constants as a way to quantify the dynamics that we are talking about here. And so in one of the final figures of that chapter, we compare how we treat dynamics using this equilibrium constant versus two other approaches that have been seen, and these rely on completely different models. So we tried to use the exact solution based on protein enzyme binding, actually, for this reversible type of reaction in order to quantify the exact concentrations that we would expect. The two examples that we compare to, one of them uses a Bell model and the other one uses a Poisson distribution for their stochastic approach. And so these are three fundamentally different approaches to treating dynamics in modeling dynamic systems. And yet when we compare how they predict the changes in a system should be, we see a high degree of overlap between them. They're relatively conserved. And so that's where we get some confidence that, the model that we're using and the models that they are using, in general terms, at least with respect to the KEQ, can be used to describe any reversible interaction across a lot of different and diverse systems. You are correct. If we were trying to talk about a model that was specifically for the K1 and K-1, yes, many more chemical or the nature of the interactions would need to be considered more explicitly. But the way that we've set up our model, I believe, generalizes it enough to simply the relative proportion of bound and unbound states defined by the equilibrium constant such that it is applicable in the broader sense to many of these diverse systems.
I would like to switch now to chapter number 6. And there I would like you to generally comment maybe what you can disclose. So when you analyze the strain stiffening properties of your different formulations of your hydrogels, and you mentioned that in one of the formulations you kind of approach the critical stresses that you will find in biological settings. So can you maybe elaborate on that, what you can say, which alternative strategies could you apply to approach or even to match the biological range?
So the biological range of strain stiffening is very common in native tissues, and it's seen quite regularly within natural biopolymer-based systems. It's very rare, firstly, to see strain stiffening within synthetic systems, and so there's been quite a bit of recent work trying to tease this apart, specifically in the context of dynamic covalent chemistry. So the work of Matthew Weber out in the States, he's done quite a bit of work on that so far. In the context of this chapter, we were quite surprised when we first saw it. It wasn't something we were looking for, it wasn't something we were expecting, but it was interesting enough that it definitely merited digging a little bit into. And given the size of that paper already, we didn't really have quite so much time and space to go do a full analysis at that time. We see in this chapter trends in terms of the changes in weight percent versus the degree of strain stiffening or the onset of strain stiffening that agree with the proposition of Weber that in fact dynamic covalent systems, even in semi-flexible polymer backbones, may have some mechanism for an inherent strain stiffening. And by extrapolating that trend, we believe that if we went to a lower weight percent and perhaps a longer cross-linker with a higher valency, that we can reasonably expect to shift that even lower if we had if we went ahead and did a systematic study in the future. Does that answer your question?
Yes, I think that's enough. So given my time, I'm looking at the Mr. Prorector. You tell me. Do I still have?
Thank you very much for your opposition, Dr.
Thank you.
And then we go to the following opponent, that is Professor Tibbet. He is Professor in Macromolecular Engineering in the ETH in Zurich, and that is in Switzerland. And thank you for coming all the way from Switzerland, albeit online. The University of Maastricht is always very glad with guests from abroad. Professor Tibbet.
Okay, thank you, Mr. Prorecker, and thank you, Mr. Candidate, for the fantastic thesis and the very nice description of it today. And I'd like to congratulate you on the really comprehensive and excellent thesis. And it's really at the forefront of the field, and it's already having impact in our lab, and I'm sure in many other labs. So congrats, and you should be proud. So I have a few questions, of course. One is that, you mentioned very nicely at the beginning that you want to quantify, and you did quantify KQ values, K1 and K-1 values. And if I recall correctly, in most of your work, you used UV-Vis and NMR spectroscopy for the majority of this. Maybe if you can comment on what other approaches would be possible, and what are the limitations and benefits of these, and maybe how happy are you with your values, or do you think we as a community need to sharpen these values in some way, or are we kind of there?
Highly esteemed opponent, thank you for your comments and your question. First off, I'm just going to start by saying ITC would be fantastic. Isothermal titration calorimetry would have been an amazing tool to have to do these measurements. That's not something we had ready access to when we were elaborating on this work. Right at the end, if we were doing any more work on this, that is definitely the technique I would want to use. And the primary advantage there, apart from its sensitivity, is that it directly correlates a thermodynamic parameter. So like in this case, the change in temperature and the change in the delta G or delta H of these systems, which can then be much more strongly tied to modeling, and where we also calculate thermodynamic parameters, for example, using DFT. That would be an amazing setup to have much higher confidence in the precision or the accuracy of these values. That said, I'm not dissatisfied with the values that we have here. If you look at the footnotes in the table where we list all of these values, you do notice that there is some aggregation where we sort of use a global fit rather than an individual fit in order to constrain some of the fitting to give us reasonable values out the other side. I think as with many kinetic experiments, the relative error that is reported for them tends to be quite large, and that's not unusual. I think I would argue that most of our values should be in the very good ballpark. I would never say that any of our values are exactly the value that describes that exact reaction perfectly. Of course, one of the downsides of using UV-Vis is that it is an indirect measurement, because that relies on the assumption that the signal that we are getting is purely from the imine that we are forming, that it has no contributions from a loss in any sort of baseline absorbance from the constituent components, that there is not a difference in contribution from the potential cis or trans isomers that can exist of the imines that we are forming. While most of these are expected to be small to negligible contributions, we have not studied those and we cannot technically rule them out. NMR, on the other hand, will directly measure the species concerned, so we have no uncertainty about what exactly we are measuring. However, the detection limits of NMR, so we did these on a 700 megahertz cryoprobe. The concentration detection limit for that particular machine with the phosphate buffer system that we use in order to regulate the pH should be around the 10 micromolar. And so then performing these at a high enough concentration in order to have a reasonable amount of measurement time with the machine in question can be practically challenging, and the treatment of that data to get consistent integration, baseline analysis, especially when the shimming may change over time, where the samples can also evolve, is a practical challenge to the implementation of that. Ideally, if we want to really nail these down, which I think as more models develop will become an increasing need, I I would start with ITC for sure, and then I would back that up with some NMR studies to just verify one or two systems, knowing exactly what species we're looking at along the way. I think that would be a very robust platform moving forward, and potentially a gold standard 10, 20 years from now. But since there aren't really any quantified systems, or very many quantified systems out there to begin with, I think we've made a pretty solid start.
No, I certainly agree with this. I think you did a fantastic job characterizing them. So I don't mean it as a criticism. The discussion was perfect. That's exactly what I'm curious about. So then maybe also you showed it on slide 6. I'm really curious about this effects that you saw where coupling some of your molecules to macromolecules. Well, you alluded to it on slide 6. I guess you didn't explicitly talk about it, but that you can affect the reactivity by coupling these small molecules to macromolecules. So whether it's the size of the linker in your network and how that affects intra versus inter, or actually the kinetic parameters that we just discussed. So maybe could you elaborate on this a little bit, and what do we as a community need to dig into to understand this a bit more? I think this is a really important point, and I think people are starting to appreciate it, but we don't have it all figured out yet. So yeah, curious what do you think about that?
Okay, so the field tends to use a relatively small subset of natural biopolymers which have a narrow degree of degrees of functionalization, some variation in their molecular weights, and we tend to use peg crosslinkers. There's a lot of unexplored space there that we haven't really looked at and that we don't really know how to leverage. So we went back to asking basic questions of how long, how many functions. We can see that excluded volume effects when two macromers are reacting, which is a specific use case for the practical systems, is actually quite important. It gives us a drop in an order of magnitude from the apparent reactivity of these systems. That's going to change the mechanics without the other side without question. And then we see when we start playing around with the valency and length of these, that actually the degree of we see big changes in mechanical properties that would align with different big changes in the balance of intra versus intermolecular crosslinking. Previous work has shown that that can be both a design parameter. Apple's lab out of Stanford did a lot of works introducing end defects to use that as a way to tune mechanics. So understanding how our choice of not just the polymer, but also the bridging or the crosslinker is going to be very important, I think, moving forward as a secondary means to fine-tune a lot of the behavior. And it also looks like it has quite a few implications in the strain stiffening behavior, which I think will be increasingly important as we nail down a lot of the more standard mechanical parameters.
Okay. Thank you.
Thank you very much.
Thank you, Professor Tibbett, for your opposition. Now we're going to the following opponent, and that's Dr. Delplace. His expertise is synthetic extracellular matrices in the INSERM Institute in Nantes, and it is in France. And thank you for coming all the way from Nantes in France.
Thank you, Mr. Dear Mr. Candidate, first of all, I'd like to congratulate you for the manuscript, which I much enjoyed reading, and I have to say it will be certainly a Bible for the next generation of students. So congratulations for this. So we only have a couple of minutes, so I'll start right away with a question, a very general question context. Apparently you've been sticking around for quite some time, so you've seen the field of bioink design evolving, so how it has changed over the last 5-7 years and how you position your materials and strategy in the current context.
Highly esteemed opponent, thank you for your comments and your question. Yes, it's been almost 7 years. Some of the major differences that I've noticed myself in this time. When I first started, rheology was not yet a staple method for the characterization of these class of materials, and that's slowly beginning to change more and more and become the baseline that everyone expects. And so a lot of the early work, the definition, if you will, the degree of quantification of these systems is often a little bit more vague than we might expect now. The other transition that we see is, so if we start from the Engler and Disher papers, early 2000s, where we first discover that, oh, cells kind of really care about how stiff the substrate they're on is. 2010, 2015, we finally see that actually the viscoelasticity, these more dynamic, these slightly less common mechanical properties also play key roles.
Sorry, my question was really focusing on bioinks.
Oh, bioink specifically, sorry. Okay. So the introduction of dynamics has been a great boon for the development of bioinks with a wider range of mechanical properties due to the inherent dynamicity allowing the printability of systems that would previously have been unprintable. The classic paradigm is that in order to provide a bioink with the necessary viscosity to be printable while maintaining desirable mechanical properties to direct or drive a certain cell response, that was a big challenge. The dynamic systems allow us to decouple to a certain extent the processability or the printability of material systems from their static or at-rest mechanical properties which we use to drive cell behavior. Over the last 5-7 years, that's definitely been one of the major driving forces, I believe, in changes in the way that bioink development is being approached within the field.
Okay, thank you. I have time. So I have one question. I think you were talking about it, biomimicry. I think this term is only used twice in the entire manuscript, and yet it seems to be the essence, the very paradigm that you're following. So do you think we know enough about how tissues, living tissues, are behaving to say this is the right direction for our research? Do we know enough about cell material interactions and so on to say that this is definitely the avenue of research to pursue?
That is a very good question to be asking. I would argue that right now we probably think we know a lot more than we actually do. We know that cells interact with these materials. We have some idea. So if you can look at the force loading rate model, the Cusack-Sachs the molecular clutch model, for example. It's very well celebrated. It manages to capture a lot of the behaviors that we can observe. And yet it doesn't capture the space that we have of integrins. It doesn't provide a method for us to characterize the force loading rate of a molecular strand of a polymer that we put in a network. It raises more questions at a fundamental level than it answers. It could be the way forward where dynamics solve all of these problems and the degree of dynamicity might directly correlate with the local chain force loading rate. We won't know until we investigate that. And as with most research, when all we have are questions, we can only pick whichever one we think is the most probable at the time and see where it takes us.
Okay, thank you. Yeah, there is a risk that this is all self-generated science, but it's interesting. Okay, 2 short questions, I think, to close the session. So, reading your manuscript, anytime you had cells, or almost every time you had cells in the system, it was on top of the gels. So, provocative question, is there any problem with your gels for cell encapsulation?
No, chapter 4, we do them in 3D as well. Final figure of the SI, I believe. We definitely printed them there in 3D and saw exactly the same observations in 2D. Most of the time, I mean, we have other projects that aren't in this thesis as well, where we have done 3D encapsulation across multiple different cell types. For the work that we've done here, the focus is really on understanding what are the primary interactions, what is viable and not viable, and what or how powerful are these tools that we're trying to create. And for most of intents and purposes, we can understand whether that's the case at this initial level by doing a 2-dimensional study, which practically is, it's quicker, it's faster, it's easier, it gives cleaner readouts. And so we predominantly start and stick with a lot of them, but no, I do not believe there's any problems with our systems for 3D.
Thank you, Dr. Delplace, for your opposition. Mr. Morgan, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. And I request that you and your company await the results of our deliberations and our return in this room. With that, I suspend the meeting.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. To the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
so, yeah. Already? I don't know.
I reopen this academic ceremony. Mr. Candidate, Mr. Morgan, the degree committee here present has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Dr. Baker is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor, Dr. Baker, to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I do so promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Francis Luke Culliford Morgan, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. I'm going to stand and take a picture and everybody clap. Okay. And I get to do the laudatio now as well. Dear family, colleagues, honored guests, and most importantly, Francis, today marks not just the culmination of a PhD journey, but the celebration of someone who has truly embodied what it means to be a pioneer in science. As just the second PhD student to join our young group, Francis arrived with what I can only describe as an unquenchable thirst for understanding for quite a lot of things, and eventually we challenged it into dynamic covalent hydrogels. Though I do suspect that none of us could have predicted just how many hours you would spend on the rheometer. Speaking of which, I believe the rheometer deserves a special mention today. Francis didn't just become proficient in it, he turned into what we might call Merlin's rheology evangelist. If you conducted any rheological experiments here at Merlin between 2018 and 2023, chances are you were inducted to what I started by calling in my head Francis Morgan's School of Rheology. His evangelism, which I can't pronounce, and teaching others was so thorough that I sometimes wondered if he was secretly writing a second thesis titled Rheology is the Best, and Here's How You Should Love It Too. With that, I'll turn the mic over to Professor Moroni, who is also your promoter during a large part of this and head of the institute.
Thank you, Matt. Just a few words from me, Dr. Morgan. First of all, actually, to thank you for all the work you have done. It was actually a lot, and also with quite a high quality and impact. I think this has been recognized by the whole committee during the assessment as well as today, for which, of course, the three of us has been quite proud also of what you have been achieving these many years. Impact and time, I think, are two characteristics also, not only of your journey, but also of our relation as a student and a supervisor. And I think I like to make also a little bit of a joke, actually, in terms of time. I think sometimes our chat in the office would sound to me like Salvador Dali kind of painting, you know, the one with the clock that is kind of losing time. Because actually Francis, and probably the friends in the audience will also recognize that as the unique characteristic to be extremely eloquent, and then with me it was always at times where I had the next meeting 3 minutes after. So I actually also would like to apologize to you today, Francis, if at times I was a bit rude in cutting off the chat, which otherwise were always extremely pleasant to have. And I think I'd like to extend also my congratulations to your family. You have a brilliant kid, a very nice chap to have around, not only as a student from a supervisor point of view, but also as a friend in the community. I wish you all the best for the next journeys. I'm pretty sure that we will be keeping hearing of all your fantastic achievements. And again, with this, I'll give back the word to Matt.
So now I get to continue. Francis's impact, though, really extends far beyond his love of rheology and his ability to eat up our time whenever we are late to a meeting. He arrived at our lab just as we were beginning to explore the space, and through his really analytical methodology and what I can really only describe as an uncanny ability to spot the significance of unexpected results, you transformed this exploratory project into really a cornerstone of our research and grew along with the field. Your work has yielded multiple high-impact publications, patents, is the foundation for one of our spinoffs, and has fundamentally challenged how we think about the role of chemistry and polymer physics in our field. Now, Francis has a particular way of approaching problems that deserves mention. When he believes something should be done a certain way, well, let's just say that moving mountains might be easier than changing his mind. What makes Francis special is not his occasional stubborn streak, it's also his ability to take criticism, process it, and sometimes after a bit of convincing and friendly debate, emerge with even a stronger approach. You're a high-performing student who have high expectations of others and yourself. I'll never forget the time when I was late reviewing one of your first papers and you got upset and you said, "What do you even do with all your time?" A little mad, took a deep breath, but then we sat down and Francis has now learned what all it takes to become a PI and how research works in the background. This combination of rigor and adaptability is rare, and it's one of the many reasons why you become a force in the lab and for the field. Coming from New Zealand via France to join us in the Netherlands, you've really embodied the spirit of a modern international science. And in true fashion, you didn't just join our community, but became an integral part of it, as we often joke, part of the furniture. You learned Dutch, joined the water polo team, had formed a climbing group, was always down for a chat. Francis, as you prepare to take the next step in your career, reminded of something quite remarkable. You really managed in our group to help create an environment where rigorous and blunt scientific debate can coexist right next to supportive collaboration. It took us a while to get there, also as a group, but really you were an integral part of that. Francis, you've been more than a PhD student. You've been a pioneer, a teacher, a challenger of ideas, and a community builder. It's not only advanced our understanding of alginate hydrogels or covalent hydrogels, but also left a mark on our institute's culture and capabilities. As you defend your thesis today, know that you've already demonstrated the qualities that make an exceptional scientist. So one of your committee members said that, a few have said that you're far beyond your years. Creativity, rigor, resilience, and the ability to bring others along the path, and the joy of discovery. Though we'll miss your daily presence in the lab, and yes, your impromptu sparring of research project ideas when we're not even really sure we even asked, we're excited to see you move to the Dresden group of Ivan Minev to combine your expertise in biomaterials to further the quest for integrating devices into the human body. They are gaining an enormous asset, and I know they realize this. Congratulations, Dr. Morgan, you've earned it.
Dear Dr. Morgan, also on behalf of the University of Maastricht, I congratulate you with the degree you have acquired, and I hope you can celebrate it in the right way, because scientifically this is probably one of the most important days in this career. I extend my congratulations, of course, to your supervisors, Dr. Baker, Professor Moroni, Dr. Bauer, and of course to your parnames, family and friends and colleagues online and on site, and I hope you all have a great day. I would like to thank the opposition, online and on site, for their work, and not in the last place, the PEDEL and the technical support team who made this hybrid session possible. Having said that, I'm about to close this academic ceremony, but I'm going to explain you how it works hereafter. We are going to ask the audience on site, of course, to go already to leave the room and go already to the rafter on the right side where the reception is. We stay here, this team, to make a photo, to make a picture with the 3 online opponents. So we would like to keep the photographer here, and the rest of the audience can go to the rafter. And then we will go to the foot of the stairs, where we make another picture. And then we go to the rafter as well. And if you want to know when you can congratulate the young doctor, then it's quite easy. You watch us, we go first, and you follow. Having said that, I close this meeting.